Now let's talk about the eukaryotic chromosome structure and the concept of ploidy. Eukaryotic chromosomes have a very complex structure, so let's start from the top. Now, we have learned that DNA is made of a double helix that is kind of attached together through hydrogen bonding with the bases on the backbone. But the chromosome in eukaryotic cells aren't present in this naked form of only polynucleotide. They're actually wrapped around a complex of proteins called histones. So this double helix wraps itself around this complex of proteins. Now, when you look at the chromosome, when you zoom out, and at each step you're zooming out, you will see that the chromosome of eukaryotic cell look like kind of these hamburger-shaped stuff that are sitting next to each other, one after another. And each of these hamburger-shaped structures is one of these things. So the eukaryotic chromosome looks like a string of hamburgers, as I said, and this is basically DNA wrapping itself around these histone molecules. So most of the lifetime of a cell, the chromosome is in this stringy type of format, which is quite loose. It's like a big piece of yarn, and it's spread all over the nucleus. Now, when the time comes for a eukaryotic cell to divide, a couple of things happen. First of all, the eukaryotic DNA is copied, which we call a process called replication. So each chromosome, just like what we saw in bacterial cells, the cell makes a copy of its genetic material. Another thing that happens is that the chromosomes are condensed. Imagine that you have a yarn that is all the way open, but then you start wrapping that yarn and making it condensed. So that's what the cell does, because it's really difficult to manage all these strings of DNA and try to evenly separate them between two dividing cells. So replication and condensation. And this condensation happens right before the cell starts dividing. And this, you'll see a structure something like this. Okay, so let's dive a little bit more into the structure of a eukaryotic chromosome. And usually we draw it with something like this. These two shapes on the top and bottom, we call them the chromosome arms. And this circle, this structure represented as a circle, which we call centromere. And this is a very important part of a chromosome, as you will see. Now, chromosomes come in all sorts of different shapes. They can have one short arm and one long arm, or they may have equal-sized arms, and they could be really tiny. So the chromosomes can be really big or really small, and also the size of the arms change. Now, what happens when chromosomes divide? Chromosomes replicate. When a copy of a chromosome is replicated, it gets an exact copy of itself that is attached to it through the centromere. That's one of the important parts that centromere makes. So what will these two like when they're replicated? They look like this. So this group of chromosomes here, this is how we would draw them for a chromosome before replication, and this, what a chromosome look like when we draw them after replication. So if in an assignment or a quiz or whatever, we ask you to draw a chromosome, you can use this type of drawing. Even though the chromosome before cell replication really is not this condensed form and it's all stringy, but for ease of representation, we're going to show chromosomes like this. So these are chromosomes before replication, that's after replication. So what do we call these copies? We call them sister chromatids. So when a chromosome replicates, it earns a sister. These sister chromatids are identical, and they are attached through the centromere. This is a display of all of the chromosomes in a cell of human being. This human being is female because it has two X chromosomes. And you can see that chromosomes come in pair. So let's take a look and see what all of this stuff means. The total complement of genes for a human being are spread across 23 chromosomes. So what does that mean? So let's go back to the example of a bacteria. So if you remember, we said a bacterial chromosome, all the genes that a bacterium needs is present on one circle. That's all it has, okay? But now imagine in a eukaryotic chromosome, this has happened. Imagine that you have this huge piece of chromosome and you're going to divide it into pieces. Okay? And then each of these pieces would get their own centromere. So this is happening in eukaryotic cells. Eukaryotic cells do not have one piece of chromosome as their genetic material. In eukaryotic cells, the genetic material is split into different pieces, and each of these is a chromosome. Okay? So that's where we get all these chromosomes. So how many pieces are there? It depends on the organism. In organisms, all the genetic components that a human being requires is all spread across 23 chromosomes. That's an important number. Why? Because we call it the N number. The N number is the total number of chromosomes that an organism has in a complete set. So the N number for humans is 23. Now, all human cells, except for sperm and egg, have two sets of chromosomes. That's why you see two of each in here. We have two chromosome 1, two chromosome 3, two chromosome 4, 5, so on and so forth. Okay? So therefore, in human cells, we have double, but in sperm and egg cells, there's only one set. So the number of chromosomes in a sperm or egg is 23. In all other human cells, because they have two sets of chromosomes, the total number of chromosomes is 46. So other human cells have 2N chromosomes, but sperm cells have N. So let's talk about ploidy. Ploidy is basically a term that is communicating how many sets of genetic material, how many sets of chromosomes one cell has. And as we mentioned, sperm and egg cells only have one set of chromosomes. We call cells like this haploid. All other types of cells in a human body, they have two sets of all the chromosomes. So therefore, they are diploid. Okay? So haploid cells only have one set of chromosomes. Diploid cells have two sets of chromosomes. And di refers to the number 2. So let's take a look and see what it looks like in numbers. So human skin cells, they're not sperm or egg, so therefore they are diploid. They have two sets of chromosomes. So what is the number of chromosomes in one set? It's 23. But since this cell is diploid, the total number of chromosomes it has is 46 because it has two sets of these. Human mouse cells are diploid. The N number remains the same because we are still talking about a human. And since since this is a diploid cell, therefore it has twice as many chromosomes. So the total number of chromosomes per cell is 46. What about human sperm? Well, we just said that human sperm is a haploid, so it only has one set. So therefore, it only has 23 chromosomes. This is true in other eukaryotic organisms. So in horse, a horse skin cell is diploid. The total number of chromosomes in one set, or in other words, the N number for the horse is 32. So since this is a diploid cell, then the total number of chromosomes in a horse skin cell is 46. What about horse eggs? The horse egg cells is haploid. The N number doesn't change because we're still talking about a horse. The total number of chromosomes per egg cell in a horse is 32. So I hope that makes it more clear. So let's go back to this one. We talked about these chromosomes that are pairwise. There are two sets. So what is the relationship between two pairs? Where do they come from? The chromosomes in a pair are called homologous chromosomes. So let's look at the chromosomes for this baby. So if we were to look at the chromosome for this baby from its cells, let's say skin cells or something, we would see chromosomes pairwise. So where do they come from? Remember, so babies are made, you have a mother and a father. So one of those homologous chromosomes come from mother, the other one comes from the father. When we go to the next chromosome, one chromosome comes from mother, the other one comes from the father, so on and so forth. So the baby skin cells has two sets of chromosomes. One set comes from the mother and one set comes from the father. So the baby has two complete sets. Okay. We call these pairwise, as I mentioned, pairwise chromosomes, homologous chromosomes. If you look at homologous chromosomes under the microscope, they look indistinguishable, but they are not identical. Homologous chromosomes carry the same genes, exactly the same genes, but they may carry different versions of the same genes. What does that mean? Let's just, for an example, think about the gene for eye color, okay? So the gene for eye color comes in variety of different versions, and we're going to look at two different versions. You could have a blue eye gene, so an eye gene that codes for blue eyes, and we have one for brown. And let's imagine that this gene sits on this yellow chromosome. Now, both the parents, chromosomes from the parents, carry exactly the same gene. Both of these chromosomes carry the gene for eye color, but the one coming from the mother has the blue version, and the one coming from the father has the brown version. So that's why we are saying homologous chromosomes are not identical. They carry the same gene, but they may carry different versions of the same gene. It is really important for you to understand the difference between homologous chromosomes and sister chromatids because it's easy to mix them up. Now, we're going to go back to these yellow chromosomes again as an example, and we're looking at the gene for eye color. These are homologous chromosomes. So this chromosome that is duplicated is homologous with this chromosome, okay? Because they have the same genes, but they have different versions of the same genes. But these two chromosomes here, these are called, we call them sister chromatids. They're attached through the centromere and they are identical. These are sister chromatids. So this is homologous to this chromosome. They have the same genes, but they carry different versions. But this here that is attached, which we call chromatid, this chromatid that is attached to this other chromatid through the centromere is a sister chromatid. So these two are identical. Sister chromatids, homologous chromosomes. So let's solve a problem. Consider these two pieces of genetic material. Imagine you're looking at them under the microscope. What are they? Let's go through each of these. Homologous duplicated chromosomes. Well, these are good since we're looking at a microscope and we say homologous chromosomes look identical. Well, that's kind of maybe, that's possibility, but it's telling us that the chromosome is duplicated. And when a chromosome is duplicated, it should have a sister chromatid that's attached to it through the centromere. So A is not correct. Sister chromatids. Again, it's tempting since these two look identical to say, oh, they're sister chromatids. Well, no, because they're not attached through the centromere. One duplicated chromosome. Well, this is actually another way of saying answer B. If this chromosome, if you had one duplicated chromosome, the sister chromatids would be present and they would be attached through the centromere. So C is not correct either. D, homologous unduplicated chromosomes. Yes, because under the microscope, these two homologous chromosomes look identical, but they don't have sister chromatids, so they are unduplicated. So the correct answer is D.